Okay, so it's time to talk about biochemicals now. There are four basic biochemicals in cells. They are nucleic acids, proteins, carbohydrates, and lipids. And the first one we're going to talk about is nucleic acids. So nucleic acids, you remember from a previous chapter, are the biological storehouse of information. They come in two forms, DNA and RNA. Remember, DNA was double-stranded, RNA was single-stranded. DNA stores biological information in cells, and many viruses, RNA stores information in some viruses. Um, so basically, what a nucleic acid is, it's a polymer of little nucleotides. Nucleotides are basically what these things are right here. Okay, this is cytosine, which is the pyrimidine nucleotide. Waiting for this to reload. There we go. This is the phosphate. This is the sugar. And this is the nitrogen base. Okay, the NH2 group out here is not here by accident. That's to allow hydrogen bonding between this nucleotide and other nucleotides on the other side of the strand, like guanine. Let's erase all that highlighting. So basically, the 5' end of the nucleotide is here. This is a carbon atom here, and that's the 5' carbon. The number 1 carbon is right here. Its purpose is to attach to the nitrogen base. The purpose of the hydrogen bonding area here is to allow this pyrimidine to hydrogen bond to a purine on the opposite side. The number 2 carbon, this is the 2' carbon of the sugar. The 2' carbon's purpose is to identify this nucleotide. If it's got an OH here, it's actually a ribonucleotide, and that's why this says RNA nucleotide here. If it had just an H there instead, that would be a deoxyribonucleotide. So it's the number 2 carbon there that determines whether it's a DNA or an RNA nucleotide. The number 3 carbon here, that's the bonding point to the 5' phosphate of a previous nucleotide. The number 4 carbon right here, that's part of the backbone. That just connects the 5' to the 3'. The number 5 carbon's job is to connect to the phosphate. It is the phosphate here that will then attach to the 3' carbon of the next nucleotide. So this goes through some of the differences here and some of the important features. The different nucleotides are due to different nitrogen bases. Adenine and guanine are the purines, and they're double ring bases. Cytosine, thymine, and uracil are single ring. We've already talked about the RNA differing from DNA at the 2' carbon. Alteration of the nitrogen base activity, nitrogen base sequence of a DNA molecule changes its activity, whether it can transcribe and translate, and ultimately the kind of protein it's going to make. The phosphates here, they do something else that's important. You see that little negative charge there? The phosphates actually impart a negative charge to the nucleotide, and that's the reason why DNA migrates from the negative pole to the positive pole. It's repulsed from the negative pole and attracted to the positive pole in electrophoresis. If you remember, nucleic acid information is read this way, from 3' to 5'. So actually, I guess like that, through the backbone. So this author drew the phosphate sticking out at an angle where it would really be kind of sticking up this way. But that's the way new information or the existing information is read. New information is always created, remember, in the opposite direction, 5' to 3'. One oddity is it's the deoxyribose, which it's not flat. It actually causes the DNA to have the spiral design that it has, and it's because these carbons actually, if you look at the molecule from the side, they zigzag. And because of the zigzagging, that's what causes the molecule to actually create the staircase shape that it has. And I think you already remember this, but the nucleic acids are directional. What that means is that direction of DNA enzymes is always from the 3' to the 5' in operation. So this molecule would be read that way, and this molecule would be read this way, and that's because this 5' phosphate sticking out here, and up here we have a 5' phosphate sticking out the other side. The RNA molecule here is different than the DNA in that it's single-stranded, and this backbone here that's got a different color has ribose instead of deoxyribose. So it's the atomic structure of DNA and RNA that determine its properties, its capabilities, the fact that it can hydrogen bond and form double strands like in DNA, the fact that RNA can hydrogen bond from anticodon to codon, that's made possible by hydrogen bonding, the fact that it has a backbone made of sugars and phosphates, all made possible by those little molecular subunits with little functional groups like amino groups and hydroxyl groups, which make it polar and capable of hydrogen bonding. Which brings us now to the next portion, proteins. Proteins are more complicated than nucleic acids. They basically are determined by their amino acid sequence. The amino acid sequence of a protein determines its 3D shape, which in turn determines its chemical behavior. Really important here, there are 4 levels of construction in a protein. The primary level is the amino acid sequence. The secondary level of construction is the coiling or the zigzagging or the folding of the amino acid chain. The tertiary structure is then a superfolding of the secondary coils on top of each other, and the quaternary structure is the union of 2 tertiary polypeptides with one another. So before I go into explaining like what all these are in terms of diagrams, this is an amino acid. This here is the amino group. Notice the nitrogen with the 2 hydrogens capable of hydrogen bonding. This is the central carbon. This is the radical group. This is how all amino acids differ by what's attached right there. This is the carboxyl group. The carboxyl group, also capable of hydrogen bonding because of this here, is the bonding area for this amino acid to the next amino acid. All peptide bonds are bonds between carboxyl groups and amino groups between amino acids. The primary structure of a protein is just these guys in sequence, the little amino acids in sequence. The secondary structure is immediately caused by these radical groups interacting with each other between different amino acids in the chain. They're like students talking to each other while in a line, say, walking through the hallway. Those interactions cause patterns, which are often coils or pleated sheets. The tertiary structure, on the other hand, is folding. Those are all, it's more than folding, though. Golgi apparatus specializes in making what are called disulfide bridges between sulfur groups on adjacent amino acids on separate polypeptides. So if we look at some diagrams now, and let's scroll down here a little bit. All amino acids differ by their radical group. The polar amino acids are serine, threonine, asparagine, and glutamine. That means that they are capable of interacting with water. Fully charged amino groups or amino acids are arginine, histidine, and lysine. Those also love to mix with water. Anything with polarity or charge loves water. Those are positively charged. Negatively charged amino acids are aspartic acid and glutamic acid. They also love water. But the nonpolar ones are these, and these are really important. Alanine, valine, isoleucine, leucine, methionine, phenylalanine, tyrosine, and tryptophan are all nonpolar, and as a result, when they are found in high number in a protein, that section of the protein will wedge into the core of a cell membrane. Really weird amino acids are cysteine, glycine, and proline. Cysteine, especially for its sulfur atoms, it's capable of forming disulfide bridges between separate polypeptides to form a quaternary structured protein. So in the diagrams here, this is the primary level of amino acid sequence, or the primary construction of a protein. The secondary construction of a protein is coiling or pleating, forming zigzag chains, which ends up forming a flat platform of atoms. This is secondary. Tertiary is when the coils and the pleats all take on a superstructure and some kind of shape caused by these groups interacting with each other and water on the outside. And quaternary is, these are 4 polypeptides, each with a tertiary structure stuck together by some kind of forces, often by disulfide bridges. This is hemoglobin right here. That's the standard shape of hemoglobin. So understand that pH, or the chemistry of the water around this kind of a protein, will change the shape of these peptides and then change the function and the ability of the peptide to operate. Polypeptides are directional. In other words, there is an amino end and a carboxyl end. So this is the first amino acid, this is the second, this is the third, this is the fourth. Each amino acid has its amino group here, 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 here. The carboxyl group, let's erase that for a second. The carboxyl group of asparagine is there, and its amino group is here. The amino group of glycine is bonded to the carboxyl group asparagine. The carboxyl group of glycine is bonded to the amino group of phenylalanine, and so on. So there's a direction to the polypeptide through the amino acids. And the reason for that is ribosomes are extremely rigid in how they bond them together. The linear sequence of amino acids was what the ribosomes put together. And it is a condensation reaction that actually puts the amino acids together during translation of protein production using DNA, which made RNA, messenger RNA, which then helped with transfer RNAs to make protein. You remember that from a previous chapter. So the linear sequence of a polypeptide is the primary structure, but no polypeptide is just a chopstick of amino acids. They always coil or pleat or loop and then fold over each other into a tertiary structure. Generally speaking, proteins that have lots of polar groups tend to mix with water well. They are free usually to move around in the cytoplasm of a cell. Proteins that have large numbers of hydrophobic amino acids end up wedging themselves into cell membranes. Those would be things like G protein linked receptors, G proteins, adenylate cyclase. We talked about all of those are important in communication. Marker proteins, the sodium-potassium pump, sodium leak channels, potassium leak channels, they all have a whole bunch of nonpolar amino acids that makes them wedge in the cell membrane. That's why they do that. The ends of all polypeptide receptors, though, have polar groups. That's why they're facing out into the solution, and they serve as receptors for other molecules to stick to. So proteins are really complicated. Proteology or proteomics is the study of protein structure, and it's really complicated, and we're only beginning to figure out the structures of many proteins now. That brings us to lipids. Lipids are different than the previous two groups. They're very nonpolar. They have often a little bit of polarity, but mostly nonpolar. So linoleic acid here, carbon, carbon, all of these little vertices on this structure, they're all bonded to hydrogens around them and to carbons around them. So there's no polarity here, and as a result, that's totally nonpolar. This is polar. So this is a fatty acid. All fatty acids have a polar carboxyl group at the end, but then the rest of the fatty acid is nonpolar. Basically, fatty acids are usually not found all by themselves. They usually bond to glycerol to form what are called glycerides. Triglycerides have 3 fatty acids bonded to 1 glycerol. Here's a triglyceride. This is the glycerol residue right here. And these are fatty acid number 1, fatty acid number 2, fatty acid number 3. So this is a triglyceride here. That's cocoa butter oil. The typical phospholipid has 2 fatty acids, and this is a phosphate group instead. There's no fatty acid here. Phospholipids differ from the others in that regard. Fatty acids bonded to form glycerides make olive oil, corn oil, palm oil, all of the oils that we cook with. So it turns out this linkage right here, right there, that's called an ester bond. That's an ester bond there, and this is an ester bond here. Ester bonds bond glycerol to fatty acids. Hydrocarbon chains of fatty acids determine fats and oils basically to be nonpolar. This little region right here has a slight polarity, but not much. And as a result, triglycerides in general are butters and things that are not soluble in water. So here's 3 different fatty acids here, only differing by whether or not they've got double bonds and in the length. So this has got a double bond in it that ends up kinking this fatty acid chain a little bit. Stearic and palmitic acid do not have double bonds, and therefore they just differ by numbers of carbons. But each oil, each triglyceride differs by the fatty acids that are stuck to the glycerol. Now that brings us to phospholipids, which are constructed this way. Phospholipids are basically diacylglycerol, not triacylglycerol. Here's the two fatty acids. Here's the backbone of the glycerols back here, and here's the phosphate group right here. There's the phosphate group. So these are three different phospholipids, and there's a little chain of atoms attached here to the end. Don't worry about that. Let's go back. That little chain right there just helps to determine the polarity of the phosphate head. So this head out here is polar, and this is all nonpolar. This mess is abbreviated as a circle with two lines attached. The two lines are nonpolar fatty acids, and the circle is the phosphate. And that's how the cell membrane is constructed. It turns out that this is entirely nonpolar here, and this is where all nonpolar substances in a cell want to hang out. And the phosphate heads out here with the choline, that's what this is called, choline. The choline residues all face either the extracellular space or the inside of the cell. So phospholipids have a very strange behavior in forming these bilayers because they have bipolar disorder, that one side of the molecule wants to be with water, the other side of the molecule hates water, and that's why they form this design where the sides that hate water face each other and don't have to deal with water. That brings us to the last major biochemical group, the carbohydrates. The carbohydrates in general are very polar and they love water. So carbohydrates are basically mostly founded on Earth of glucose, fructose, or galactose, and the most common monomer on Earth basically is glucose. Glucose is full of hydroxyl groups. Here's hydroxyl group, hydroxyl group, hydroxyl group, hydroxyl group, hydroxyl group. Those 5 hydroxyl groups make it extremely soluble in water, and even polymers of glucose are extremely soluble in water. The hydroxyl determines most of the chemistry by hydrogen bonding. Carbohydrate monomers are referred to as monosaccharides. Again, that's glucose, fructose, and galactose. Those are isomers of each other because they all have the same basic formula of C6H12O6, but they have different structures. This is glucose. This is maltose. Glucose bonded to glucose forms maltose. The bonding pattern of the glucose monomers determines what the polymer will be in a glucose polymer. This is a glucose polymer. Glucose, 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 glucose. These are alpha 1,4 linkages. What that means is that this is carbon number 1, and this is carbon number 4. Carbon number 1 of one glucose bonded to carbon number 4 of the next. And they're all bonded in a regular way, and that's what makes up starch. Cellulose has a beta 1,4 pattern where the glucose molecules are inverted. The second, fourth, the even glucose molecules are inverted, and as a result, it has a different chemistry than starch, and it can't be digested by the human body like starch. So a different bonding pattern, it's called beta 1,4, makes up cellulose. And the arrangement of the bonds between glucose determines whether or not we can turn the molecule into clothing or we can cook it and make it into pasta. The orientation of the glucose subunits is basically part of the secondary structure of a carbohydrate, and the orientation of the glucose subunits determines the secondary structure. So starch is a molecule based on alpha glucose. Cellulose is based on a polymer of beta glucose. The only difference between alpha and beta glucose is this last hydroxyl group. In beta glucose, it's facing up. And as a result, that causes the molecules to flip in their pattern back and forth. Let's scroll down here for a second. This is glycogen here. Glycogen, each one of these little tiny structures that this artist drew here, let's select the pen. That's a glucose there. Glucose, 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 glucose. There's hundreds of them. And you see how there's a protein in the center, and then attached to the protein, attached to the protein, we have little glucose molecules, and they're all built out and branching in all directions, making a giant fuzzball. This is what glycogen droplets look like on the molecular level in your liver. At the core is a special core protein that glucose subunits bind to, and then enzymes attach glucose subunits in linear and branching patterns to make a big glob of glucose. This is a nice storage form of glucose in a cell. The molecules are all held down and held into position, and if the blood level of glucose starts to fall, enzymes will cut these little glucoses off and then release them into the bloodstream, which will then bring your blood sugar back up. On the other hand, in cellulose, let's erase all that. Cellulose. In cellulose, the glucose subunits are beta. So in cellulose, the molecules zigzag back and forth. And so one molecule is right side up, the next one's upside down. The next one's right side up, the next one's upside down. And as a result of this zigzagging pattern, your glucose, why does that keep happening? Your glucose subunits can't be broken apart by the enzymes in your digestive tract. So this polymer is useless to you. If you eat a cotton rag, you cannot digest it because you do not have the enzymes to break the glucose molecules apart. Glucose, 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 glucose. But you do have the enzymes to break these glucose molecules apart. On the other hand, if a goat eats a cotton rag, in its digestive tract, it has bacteria that can actually release enzymes to break those beta glucose molecules apart. And as a result, the beta glucose molecules can be used by the goat to burn in glycolysis and then send through the Krebs cycle in the ETS to process for energy, to make ATP. We can use beta glucose, but we can't use it when it's in cellulose. I should point out that a researcher has just successfully managed to industrially produce cellulose, or turn cellulose into xylose, which is a hexose, or it's a sugar, I should say. It's just a sugar, which then can be turned into either food or biofuel, which is something we've been trying to do for eons, but now have just achieved it in the 21st century. That brings us then to the next group, or the next topic, which is cells and organelles, and I'll do that in the next lecture.